For those of you who don't know me, my name is Chris. I'm one of the genetic counselors here working at Children's Hospital of Philadelphia. I've worked very closely with Dr. Skraban and previously worked with Dr. Deardorff and Katie before they've moved on to different roles at different institutions. So as part of the work that we do here, I consider it a tremendous privilege to participate in the care of many WDR26 families here in the Philadelphia area. And it's been wonderful to be able to connect with families coming from afar as well. So thank you for the privilege. I just felt that, especially given that a lot of the talks today, especially when we get into some of the more kind of science-heavy content, that we want to make sure we set the table a little bit with a little bit of a refresher of some of these genetics concepts, just so that we're all speaking the same language to a certain degree, okay? So it all really starts here, right? These are chromosomes. This contains everything in terms of the genetic material within the nucleus of the cells of our body. And as Dr. Skraban mentioned, that we have 23 pairs of our chromosomes, right? We've all, I think, heard this in many different iterations and forms at this point in time. And some people in this room have had this kind of testing done, you know, looking at chromosomes as kind of part of your diagnostic odyssey with your children, starting commonly with looking for changes within chromosomes from a broader perspective. And so this is literally what it looks like when we open up cells and look at chromosomes under a microscope. This is just the chromosomes all paired up. So we have 46 chromosomes in total. And I don't know if I actually have an arrow, but we'll see if that shows up. The WDR26 gene is located on this first pair, chromosome number 1. And just to appreciate again how much genetic material that we all carry within every cell of our body and the overall scale of them, we have this kind of mess, this purple spaghetti of all of our chromosome material within the nucleus of the cell. And if we zoom in to the closest level, we see this double-stranded helix. This is the DNA structure that literally encodes the information or these genes that are the recipes that tell our bodies how to grow and develop. This is the most complex looking slide I'm going to put in front of you, I think. I'm sorry. And this is just a cartoon showing that these are the actual letters, the 4-letter alphabet of our genes, the A's, T's, C's, and G's that we hear about in the context of genetic changes that cause differences in our health and development. Some terms, again, and just to get familiar, we've talked about, again, 46 chromosomes in total. We have about 20,000 different genes. This is an estimate. Numbers will vary that tell our bodies how to grow and function. We call them recipes, blueprints, things along these lines, because the genes are used to create proteins. And so we use these instructions to create proteins, and proteins have very specific jobs in our body. They may do something, interact with other proteins within the cell or other genes within the nucleus. They may leave the cell and interact with other cells in the body or tissues or things like that. Getting a bit of an echo. I don't know if you guys are getting that. Okay. Good. Okay, perfect. And then if you look on the right-hand side of the screen here, this is showing a gene structure. And just to get some of the terminology out there again, we may see terms like exons and introns when we're talking about kind of the structure of genes when we look at them closely. And many of us have undergone this testing, whole exome sequencing. And what the exome is really only a small sliver of our entire genome, which a genome is all of our genetic code. But only about 1 or 2% of our genome directly encodes for protein. And so we refer to that protein coding region of our genetic material as our exome. And so that's what whole exome sequencing is sequencing that 1 or 2% of the entire genome. And it makes some intuitive sense, right? You know, we target analyzing these protein coding regions because if there's a mutation or a change within that 1 or 2% of the DNA that makes proteins, that's going to directly impact the normal function of those proteins that gene encodes and can cause a downstream difference in, again, health and development in different ways. Our genes, our cells have this amazing mechanism of taking that blueprint, taking a copy of it so that the gene material itself stays protected and nothing happens to it in the nucleus. It'll take a copy of that, and then it'll refine that copy and then use that copy to provide the sequence of amino acids that actually make up the strands of the proteins themselves. I like to think of an analogy. I don't know if we have lots of cooks and bakers in the crowd here, but I find if you're trying to look up a recipe online, I find they're a lot of time bloated with like ads that you're trying to scroll through, and that cook's journey on, oh, I tried this and my family loved it, and I, highly recommend it, things like that. That are great to hear, but I need to get to the, the instructions, right? I need to know the ingredients. I need to know how much, how long to bake it, all that stuff. And so what our cells do, you can think of that as like the article, right? With that recipe. I'm going to go through that. I'm going to pull out the stuff that I need, and I'm going to get rid of the stuff that I don't. And then I'm going to use that kind of refined recipe as my reference for when I'm actually in the kitchen cooking it. And so the introns are the regions that our cells will take out of that copy of the recipe, and then it'll use just the exons to actually use as the reference to make the protein itself. Okay. And what this slide is really just showing, again, is it's kind of referred as the central dogma in genetics, right? Where we take the gene, that's the instruction, we make a copy, that's called an RNA molecule. So that's, again, kind of your copy of the recipe. The protein is what that recipe is used to directly create. And then those proteins, again, go off and do various jobs and develop the structures of our cells and tissues of the body. And ultimately, at the largest scale, we have, in this case, humans and people. So I want to ask, does anybody have a potential guess maybe about how much DNA we actually share in common with each other as humans? No, Kara, you can't answer. 99? No, you guys are too smart. Okay. So over 99%. Right? All of our genetic code, if you take any two random people and you compare the sequence of their DNA, 99.9% of that DNA will be the same between those people, okay, anywhere in the world. So 0.1% of the DNA is what actually contributes, at least highly in part of what makes us literally unique as people and the populations that we come from. And so when we're doing genetic studies and in a context in a hospital or clinic, what we're trying to find are rare changes within the DNA. Okay, this slide is just showing, if you look at the bottom, there's kind of individuals, and it's showing that most of their DNA, the sequence of it is the same when you compare various individuals, but we are all going to have natural variation within the DNA, again, literally making us unique. Most of that variation is common, and what we're looking for are rare changes in the DNA, and changes in the DNA that actually disrupt the normal function of that gene and the protein that it makes. Okay, so again, 0.1%, that's all it takes. Okay, I also just want to emphasize that many of us in this room have also heard this phrase de novo, which comes from Latin, and it literally means of new. And so highly prevalent in WDR26 are de novo changes in the DNA. In other words, that were not inherited changes of the genes that we inherited from either of our two parents. It's important to know we all carry de novo changes in our DNA. All of us have about 75 de novo changes within our DNA that didn't come from either of our parents. But the vast majority of these changes don't alter any kind of genetic function, just due to chance. But rarely they can occur in a critical region of a gene that can alter its function. These changes, again, happen spontaneously due to chance, and we all carry them, which is important to understand that that is normal. Okay. And as I mentioned, when we're doing diagnostic testing, we're looking for rare changes and not common changes. And sometimes it's not so easy to distinguish those two things. Just to go over some types of mutations that can occur within genes that can cause genetic conditions. Some of us know this term missense changes, right? So missense mutations or pathogenic variants. This is using an analogy where, again, if we look at the recipe of itself within the gene and just think of it as a sentence and take away the A's, T's, C's, and G's for a moment, what our cells do is they look at that code in kind of 3-letter increments at a time. And they use those 3-letter segments to determine what kind of amino acid is needed as the next kind of bead in the string of the protein, okay? And so this is an example of a missense change where you can have one letter that's replacing the common letter that we typically see there, and that one letter change can alter the amino acid that is required at that position in a protein. And in some cases, missense changes can then alter protein function as a result. We can also have nonsense changes. So what happens here is the kind of sentence of the gene is disrupted. It stops or halts somewhere within the gene itself. These can happen early on in the sentence of the gene, they can happen in the middle, and they can happen at the end. And sometimes there can be associations between where these disruptions occur and what that can do in terms of what that looks like in its presentation of the diagnosis. We can also have insertions. So as it implies, there are sequence variants that have been introduced into the sentence of the gene that aren't typically there. And these can kind of throw off that kind of reading frame. We refer to it as these 3-letter blocks, and can also disrupt protein function. And then we have deletions. And so again, this can be deletions of sections within a gene or towards the ends of the gene itself. This slide is kind of reemphasizing that central dogma, and again, how DNA makes RNAs, and RNAs are used to create these beads on a string that create the proteins themselves, these beads of amino acids. And those kind of fold up in a complex way to create a protein itself. And that's what that rainbow colored kind of bird's nest is the actual protein itself in this case. This is showing how when you have certain mutations that create that nonsense or frame shift, where within the sentence of the gene, there is a mutation that essentially tells the machinery in the cell to actually stop at that position. And so to no longer continue reading the rest of the gene and making the rest of the protein molecule itself. What this can do is there's actually a feedback mechanism that can happen in cells where they can actually detect when these changes are present in genes. And they recognize that this can actually be harmful, that if this protein is created, it could actually do different things that it's not supposed to do and disrupt other pathways within the cell. And so it will actually detect this and then go back and target those RNAs to say, I'm going to break these down so that I don't make protein from this. We can also see this with some types of splice site changes, which we see in WDR26 as well, splice site mutations that can also sometimes have this feedback mechanism. And the result, as you can see, is that we don't actually get any protein expression at all. And this is just showing missense changes where you can still get a protein product, but you may have kind of an area of the protein at what we call functional domains that actually can't perform its normal job. So you still have full protein there, but it might not be working the way that it's supposed to. Okay. And we've seen that within WDR26, this is caused by mutations that cause what we call a loss of function of the normal function of the gene. And this is comparing on the left, kind of this normal process of DNA making protein. And we can see this loss of function where a mutation within the gene causes reduced levels of protein as a result. And this will be something that Dr. Deardorff will add more context to. Okay. So there's obviously things that we can learn on every level. And I think that that's something that we want to always keep in mind, right? That research isn't just looking at genes. It's everywhere along the process, right? We can do research at the DNA level. There's people that research RNA. There's people that research proteins, cells, and then finally people themselves. And we also have to do research to learn what is the downstream effect of these types of mutations and what can we learn in terms of all the various unique ways that this diagnosis can present itself. So just keep that in mind as you're listening to some more of the kind of research-based talks today, okay? And I think that's everything I have.